Hello student, I am Dr. M.H. Maulavi. Today presenting Vannusals molecule. Vannusal is a triterpene, so it is very important to look at the biogenesis of terpenes. How the terpenes are formed in nature, it is a big question. So let us look at the building blocks for the terpenes. Just like proteins, amino acids are the building blocks for proteins. Here the isoprene unit, that is 2-methyl-1,3-butadiene, is a building block for all the terpenes. For example, we know carrot, and carrot contains the coloring matter as a beta carotene. This isoprene unit contains the 5 carbon atoms, and it is very important to look at the hidden isoprene unit in beta carotene. This is first unit, second, third, fourth, fifth, sixth, seven, and 8. So beta carotene contains 8 unit of isoprene. Each isoprene unit contains the 5 carbon atoms. So accordingly, beta carotene contains 40 carbon atoms. How isoprene is formed in the nature? This is very big question in nature. But student, remember that isoprene is not directly involved in the biosynthesis of the terpenes. Rather, the chemical equivalent of isoprene is isopentylene pyrophosphate, that is IPP, and gamma-gamma dimethyl allyl pyrophosphate, that is DMAPP. Both are formed in nature by the mevalonate pathway. Acetyl coenzyme starting material for the mevalonate pathway. After the series of enzymatic reaction, this acetyl coenzyme is converted into mevalonic acid. Further, after the several steps, mevalonic acid is converted into IPP, that is isopentylene pyrophosphate, and this isopentylene pyrophosphate is isomerized into DMAPP, that is gamma-gamma dimethyl allyl pyrophosphate. So these are the two chemical equivalent for the isoprene unit. One is IPP and second is DMAPP. Both contain the 5 carbon atom, and IPP is isomerized into DAPP. So these are the two important precursors for formation of the all terpenes. Let us look at the different precursor for formation of the terpenes. There are different types of terpenes: hemiterpenes, monoterpenes, sesquiterpenes, diterpene, cisterterpene, and the triterpene. Hemiterpene is formed from the isopentylene pyrophosphate, that is IPP, because both contain the 5 carbon atoms. IPP contains 5 carbon atoms, hemiterpenes contain the 5 carbon atoms. Monoterpenes contains the 10 carbon atoms. Geranyl pyrophosphate, GPP, contains 10 carbon atoms. So GPP is a precursor for formation of monoterpene. Farnesyl pyrophosphate, FPP, contains 15 carbon atoms. In nature, sesquiterpene also contains the 15 carbon atom. So we can say farnesyl pyrophosphate is a precursor for the sesquiterpenes. Geranyl pyrophosphate, GGPP, contains 20 carbon atoms. Diterpene in nature contains the 20 carbon atom. So GGPP is precursor for the diterpenes. Cisterterpenes contains the 25 carbon atoms. So geranyl farnesyl pyrophosphate, a 25 carbon unit, is a precursor for the cisterterpene. And lastly, squalene, 30 carbon unit, is a precursor for the triterpene, because all the triterpenes contain the 30 carbons. Construction of the precursor and the terpene. We have seen that mevalonate pathway gives IPP and the DMAP. This IPP and DMAP gives the higher precursor, which in turn responsible for formation of higher terpenes. Like IPP is a precursor for the hemiterpenes, both contain the 5 carbon atoms. Then GPP is a precursor for the monoterpenes, DPP contains 10 carbon atoms, and this GPP is formed by the combination of IPP and DMAPP. Then FPP contains the 50 carbon atoms. FPP is a precursor for the sesquiterpenes. And FPP is formed by the combination of IPP and the GPP. GGPP, geranyl pyrophosphate, contains the 20 carbon atoms and it is a precursor for the diterpene. GGPP is formed by the combination of IPP and FPP. Geranyl farnesyl pyrophosphate, GFPP, contains 25 carbon atom. It is precursor for the sister terpenes. GFPP is formed by combination of the GPP and FPP. And lastly, squalene contains 30 carbon atoms. It is precursor for all the triterpenes and squalene is formed by the combination of two FPP unit. So IPP and the DMEPP are the smaller units for the all terpenes, but they give rise to higher precursor and these higher precursors are responsible for the formation of higher terpene. So this is the basic knowledge about the terpenes that we should know in order to understand the biogenesis and the structures of the Vannusal A and the B. Vannusal, there are two molecules, Vannusal A and the Vannusal B. Both differs in this R group. This Vannusal A contains one extra acetate group, while the Vannusal B contains only one acetate group. Vannusal A and B are isolated from the marine ciliate, euploids Vannus, and these two are triterpenes because they contain 30 carbon atoms. This is the structure of Vannusal A and Vannusal B. The chemical formula for the Vannusal A is C34H48O8, while the chemical formula for the Vannusal B is C32H46O7. Vannusal A contains two acetate group. Vannusal B contains one acetate group. Neglecting the acetate group, it is very clear that Vannusal A and B contains 30 carbon atoms. So because of this, Vannusal A and B are triterpene. Structurally, Vannusal A and B show 6 rings and 13 asymmetric carbon atoms, out of which the 3 are quaternary asymmetric carbon atoms. It is the biogenesis of Vannusal skeleton. Two biogenesis were proposed, biogenesis A and the biogenesis B. In biogenesis B, it is conventionally supposed that this Vannusal A and B contains 30 carbon atoms. It is a triterpene, so it must be originating from the squalene 30 precursor after the series of steps. So based on this assumption, based on the formation of and the breaking of bonds in squalene units for the rearrangement and the formation of the Vannusal skeleton, this numbering is proposed. Number 1 carbon, second, it goes on like this. So Vannusal A and B contains 30 carbon atom. So it is a triterpene. Biogenesis B. Biogenesis B is proposed by the Guella and his co-workers in 2007, and it is based on the assumption that Vannusal A is formed by the dimerization of hemivannusal. Hemivannusal contains the 15 carbon atom, so it is a sesquiterpene. 2 units of hemivannusal undergo the dimerization to form 30 carbon atom unit, that is the Vannusal A and the Vannusal B. This is the mechanism for biogenesis B. Here, 2 units of hemivannusals, 15 carbon atoms, undergoes 2 aldol condensation reaction to form a basic skeleton, that is 30 carbon atoms. After one more aldol condensation reaction and the dehydration reaction, the new 6-membered ring is constructed. So in this sense, this hemivannusal contains the 6 rings. After the series of steps, it is converted into the Vannusal A and the B molecule. So this is very interesting molecule in nature which contains the 30 carbon atom. So it is a triterpene. In further lecture, we are going to look at the structure and the synthesis of Vannusals.